So, hello everyone. Today we are going to complete all the antifungal drugs. Okay. So, you have to complete all the antifungal drugs. You have to understand one very important point that. One second, let me fix my. Hey, Matisna Mabrur. ব্যাংক গীতা মানুষ আইন না বুঝে রাগ ওঠে এখন কইছি সারাদিন ওলা লাইভ হইয়া বই রুম 24 ঘন্টা এমনিও আস্তে আস্তে নানি মানুষ আবার শুরু হইবা বহুত মাথা খারাপ হইয়া আসছে আমার ফার্স্টে বেলা বালিও সুন্দরে ভিউজ আইছে সাবস্ক্রাইবার আইছে সব তো হয়েছে এখন কিতা হয়েছে কি জানো আমিও নিজেও জানি না কোনটা নি কিতা হয়েছে So hello everyone. Today we are going to complete the antifungal drugs. Before completing the antifungal drugs, I'll tell you a very important point, and that is what are the different targets. So on which we will use our drugs as an what it is called quality lab. Ota koriya khan sir. ভাই মাতিস না রে বেটা আমি রাইত সুন্দর করে পড়াইছি রাইত একটার সময় আচ্ছা কোয়ালিটির ভিডিও বলা করিয়া বানাইছি মনে করছি মানুষ আইবা ওলা সুন্দর করে পড়াইছি বিজত নিজে পড়িয়া প্রিপেয়ার ইয়ারে দে পড়াইতে লাগছি 1 ঘন্টা লম্বা ভিডিও ভাই একটা তো মানুষ নাই মাথা মগজ নষ্ট হয়ে গেছে বহুত রাগ উঠেছে আমার এখন কইছি ও নানি সারাদিন ওলা আমি আমার বইয়া বই পড়িমু আর ক্যামেরা অন করে থই দিমু এমনিও আইজ না খাইলে একবার না একবার তো মানুষ আবার শুরু হইবা আস্তে আস্তে বিচ্ছত নি ও ও ও হইছে কথা একটা কি গুলাইব আইতো কেন কেন ঠিক আমার মনে আইছে একবার কিন্তু রাগ উঠি যায় রে ভাই তুমি জানোস না এমনি আস্তে আস্তে সব ঠিক হইব বুঝছেন এখন আমি সুন্দর করে বোর্ডও নিয়ে কানেক্ট করতাম কানেক্ট করিয়ে আস্তে আস্তে ওখানে দাঁড়াতেই শুরু করতাম না আবেদ ভাই আবার শুরু করছি না পড়া নাকি তা জানি আমি তোমার সেট আপ দেখাই ও দেখো ও না সেট করছিলাম সব ওকে করিয়া ও দেখো বোর্ড উড কিন্তু প্রবলেম হয়েছে ইয়া সেট আপ বানাইছিলাম ফড়াইবার লাগে বাট অন না বুঝছো আমি দিনও সময় পাই না পারি না আর রাইত ফ্রি হইয়া ক্লাস লোয়াত লাগি যে সময় এই সময় আধা রাইত থাকে মানুষ আইন ওন মৌরুরে হয় এই নয় যেলা আমি ক্যামেরা অন করছি তা হইতো না সুন্দর করে সেট আপ লাগাইলে আস্তে আস্তে মানুষ হইবা আইলে এমনি ফড়ানি শুরু করলে এমনি সব হইব বলে কইলাম হয় এখান ঠিক কইছে আচ্ছা আমি ইনস্টাগ্রামও জিও করছি দেখলানি ইনস্টাগ্রামের আমার ভিডিও এডিট দেখলানি কিলা হইছে নি ভালো হইছে নি রাগ না বে দূর বেটা বোগা আমি ওখানে তো আর তুমি মানে বালা বুদ্ধি রে ই অফলাইন হয়ে যাই মরে ফেল মাথা শুরু করো যেদিন অফলাইন হয়েছে বুঝছো ভিডিওটা বন্ধ করে দিন তখন কি আচ্ছা না না আমার ইনস্টাগ্রামর আমার ওগিন দেখলানি ওগিন দেখো বা দেখিয়ে তোরা রিভিউ দিও সাইন কি লাগছে লাস্ট ভিডিও যে গুতে ওলা বানাইব এখন তো মাতে সে গ্যাস কে তো ক্যামেরা ওলা যদি না নি ওই যেন মৌরে আইয়া ওলা মেসেজ করতো দিন রে বা আমি লাইভ হইলে তবে কম হইলে মনও ভালো লাগলো মানে ওই বাই ঠিক আছে যা বা ওলা সারা দিয়ে দেখা যায়নি হয় দেখা যায় So hello everyone, today we are going to start the antifungal drugs in medicinal chemistry. Antifungal drugs means drugs which are used to inhibit the growth or kill of the fungus, right? Yes or no? So in these antifungal drugs, we will study all the different classes of drugs. Along with that, we will also study, first of all, we will study the targets. Targets means what are the different targets that used for to kill the fungal. Alright, is it visible or not? Yes, visible. So let's draw a fungal cell and then we will show you. So then we will see what are the different targets that are visible. So first of all, this is the plasma membrane, right? This is the plasma membrane. Along with that, we will see the cell wall. Is it visible or not? Yeah. So see, first of all, this squalene will be converting into squalene epoxidase. Squalene epoxidase. In the presence of enzyme squalene epoxide. Then this squalene epoxidase will convert into lenosterol. This lenosterol will convert into ergosterol. And these ergosterols are the component which are present. These ergosterol are the component which are present in the cell wall. Right? Like this in like this. Okay. Now the thing is that what are the different classes of drugs? So the classes of drugs are mainly based on their targets. For example, you will see in the classes of drugs of in the classes of antifungal drugs. First of all, you will see antifungal antibiotics. Is it visible? Yes, antifungal antibiotics. In these antifungal antibiotics, first one you will see is polyene. Hello. Good evening. Hello. Am I audible or not? Good evening. Hello. Am I audible or not? Good evening. Hello. Am I audible or not? So today we will see the different targets which are used in the case of antifungal cell. So number one will be cell membrane. Cell membrane, why? Because fungi and mammalian cell both contain a cell membrane that serves and play an important role in cell structure, in cell division and cell metabolism. Okay. The complex lipid particle called sterols account for the approximate 25% of weight of the cell membrane. Okay. So where are the sterols present? These are the cross-link structures. These are the sterol. So in the case of human mammal or in the case of animal, it is cholesterol. But in the case of fungi, it is ergosterol. Right? So what are these? These are. So these are ergosterol. Now, next point is that the mammalian cell contains cholesterol as sterol component, whereas fungi contains ergosterol, which is important for proper functioning of many cell membrane. Okay. So in the case of animal, these cross-link are cholesterol, but in the case of fungi, these are ergosterol. So there is a difference between sterol component in the case of animal and in the case of mammal. So we can target this in the case of fungus. We can target this ergosterol as a target component, right or no? So if we can block or if we can inhibit or if we can break down that ergosterol by using any kind of drug or medicine, then what will happen? We can inhibit the growth of the we can inhibit the growth of the fungi. Yes or no, right? So this is target number 1. You can write that target number 1. So what are the drugs that are used in the case of inhibition of sterols? Can anyone tell me? Amphotericin B, nystatin and natamycin, that is polyeins. So polyeins are used in the target 1. In target 1, what are used? Polyeins are used. So what are the example of polyeins? The example of polyeins are amphotericin B, nystatin B and natamycin. Amphotericin B, nystatin and natamycin. You have to revise. 1 second please. Let me. Yes, I think now it is fixed well. So amphotericin B, nystatin and natamycin. Okay. Next, number 2, that is microtubule. These microtubules. So these microtubules are also considered as target number 2 for cell division. So these microtubules are inhibited by using griseofulvin. That is grisio. So this griseofulvin is an antifungal antibiotic, right? Griseofulvin is an antifungal antibiotic. Next, the DNA. So this DNA are also used as another target. Target number 2. This one and this one is target number 3. And this DNA are used as a target where flucytosine are used. What is the drug? Flucytosine. Flucytosine is the drug used against DNA synthesis. So if we can inhibit DNA synthesis, what will happen? Ultimately we can inhibit the replication or division of the fungal cell. So ultimately we can inhibit the growth of the cell, right? Next, in the case of squalene epoxidase, see, this ergosterol is the main component of fungal cell wall, right? The sterol component in the fungal cell is ergosterol. So we can inhibit the synthesis of the ergosterol by using 2 different targets. Number 1 is the enzyme which converts lenosterol into ergosterol. So what is the enzyme? The enzyme which converts lenosterol into ergosterol. What is that enzyme? The enzyme is lenosterol 14A demethylase. What is the enzyme? Enzyme is lenosterol 14A demethylase. If we can inhibit that enzyme, then also we can inhibit the synthesis of ergosterol, right? So this enzyme is used as target number 4. And what are the drugs used in that target? The drugs which are used are azoles. What are the drugs? All the azoles. Can you please tell me the example of azoles? What are the example of azoles? Clotrimazole, iconazole, butanazole, miconazole, ketoconazole, fluconazole. All the different types of azoles are used to inhibit the lanosterol 14A demethylase enzyme. If we inhibit that enzyme, ultimately we can inhibit the synthesis of the ergosterol. Also, so write the examples. What are the examples? Clotrimazole, next miconazole, next fluconazole, next voriconazole, next tarconazole, next itraconazole, and etc. etc. So many other azoles are there. So by using the azoles we can inhibit the lanosterol 14A demethylase, and if we can inhibit that enzyme we can easily inhibit the synthesis of the ergosterol. And if we can inhibit the synthesis of the ergosterol, we can easily inhibit or we can easily phase or interfere with the cell wall or the moiety of the cell wall. Once the cell wall moiety is destroyed, what will happen ultimately? The cell wall will be destroyed easily, and by this way we can inhibit the growth of the fungal cell. Next, also this squalene epoxidase is an enzyme, and we can use that squalene epoxidase as a target also. If we can inhibit the squalene epoxidase enzyme, then also we can easily inhibit the ergosterol synthesis, and then ultimately it will lead to the inhibition of the growth. So what are the drugs that are used in the inhibition of squalene epoxidase enzyme? That are allylamines. What are the drugs? That is target number 5. And what are the drugs? The drugs are allylamines. Can you please tell me the example of allylamines? What are the example of allylamines? Anyone? Anyone? Neftifine. Example of allylamine? 3. Number 1 is neftifine. Number 2 is terbinafine. Neftifine, terbinafine, and tolnaftate. Neftifine, terbinafine, and tolnaftate. So neftifine, terbinafine, and tolnaftate. Now you can easily understand that the antifungal drugs, okay, let me rub all this and write it clearly and easily so that you people can understand. So there are 5 targets used in the fungal cell to inhibit the fungal cell wall growth. Okay, so what are they? Target number 1. Target 1, that is cell wall. In cell wall, which one of the following is most important component of the cell wall? That is ergosterol. Okay. So first of all, which drug is used in the cell wall? That is amphotericin B, or simply you can say polyenes. This cell wall is targeted with which drug? Polyenes. And what are the example of polyenes? The example of polyenes are amphotericin B, next nystatin, and next natamycin. Amphotericin B, nystatin, and natamycin. Next, target number 2. Target 2. What is target 2? Target 2 is synthesis of ergosterol. If we can inhibit the synthesis of ergosterol, we can easily inhibit the growth of the fungus. Yes. So synthesis of ergosterol again can be inhibited by using two targets. Target number 1 is squalene epoxidase inhibitor, and target number 2 is latanosterol 14A dimethylase. So synthesis of ergosterol is done in two ways again. So target number 2 and target number 3 is also there. Okay, so target number 2 is squalene epoxidase inhibitors, and target number 3 is latanosterol 14A dimethylase. Right? So what are the drugs used as a squalene epoxidase inhibitor? The drugs used as squalene epoxidase inhibitor are allylamines. What are they? Allylamines. Can you tell me the example of allylamine? Terbinafine, tolmastine, and 3, right? Number 1 is terbinafine. Should I write in this way? Okay, I'll write here. Number 1, terbinafine. Number 2, tolmastane. Terbinafine, tolmastane, and number 3, naftifine. Terbinafine, tolmastane, and naftifine. These are the 3 drugs which are called squalene epoxidase inhibitor, and they are also called allylamines. These are together called allylamines. Allylamine. Right? So you are what are the drugs? Polyene. And the example are, how many examples of polyene are there? 3. What are they? Number 1 is amphotericin. B. Number 2 is naftifine. Amphotericin B. Next, nystatin. Next one is natamycin. Right? Next, target number 2. Already I have shown in the diagrammatic view. Okay? Target 2. What is target 2? Squalene epoxidase. Squalene epoxidase enzyme, right? And in that squalene epoxidase enzyme, what are the drugs used? They are allylamines, right? They are allylamine. Allylamine. And what are the example of allylamines? terbinafine, tolnaftate, naftifine. Right? Terbinafine, tolnaftate, naftifine. Is it visible or not? Next we will see target number 3. What are the example of target 3? Target 3 is latanosterol 14A demethylase. Latanosterol 14A demethylase. And in that, what are the drugs used? They are azoles. Drugs called azoles. Azoles. Can you please tell me the example of azoles? Yes, so many azoles are there. Cotrimoxazole, miconazole. Right? So actually one day I remembered a trick, and that was CM, next FBIP, and next pass. Okay, so in the case of CM, clotrimazole, and miconazole. These are comes under one class of drug. Next, FBIP. F for fluconazole, next B for voriconazole, V for voriconazole, I for itraconazole, and P for parconazole. Again next ketoconazole, sulconazole, right? Ketoconazole, sulconazole, fluconazole, already I have written, right? Itraconazole, already written. Tarconazole, yes, tarconazole. Etc. So this is target number 3. Next, target 4. So what is target 4? That is microtubule. Target 4 is microtubule. Target 4 is microtubule. And what are the drugs used in that against that target? The drugs is griseofulvin. What is that? Griseofulvin, right? Am I visible? Yes. Audible or not? And last one. So that is a type of antifungal antibiotic. That is a type of antifungal antibiotic. And last target, that is DNA. Target number 5. That is DNA. And what is the drug used in that case? Flucytosine. What is the drug? Flucytosine. Right? Then flucytosine comes under nucleoside antifungal antibiotic agent. That is called nucleosides. Nucleoside antifungal agent. Okay. So simply, antifungal drugs are used to treat or to inhibit the growth of the fungal cell, or to kill or to inhibit growth, right? And these drugs are used in 5 targets. In that, target number 1 is cell wall. Target number 1 is cell wall. Already I have shown you in the diagrammatic view earlier. In the cell wall, polines are used, and the example of poline are amphotericin B, nystatin and natamycin. Next, target number 2. And what are the target number 2? Squalene epoxidase enzyme inhibitor. And the squalene epoxidase enzyme inhibitor are L-iramine. Example of L-iramine are terbinafine, tolnaftate and neftifine. Terbinafine, tolnaftate and neftifine, right? Next, target number 3 is our latanosterol 14A dimethylase. Latanosterol 14A dimethylase are target number 3, and in that the drugs are used are azoles. And example of azoles are cotrimoxazole, miconazole, fluconazole, voriconazole, itraconazole, proconazole, ketoconazole, sarconazole, tarconazole, etc., etc. So many azoles are there, right? And next, target number 4. And what are the target number 4? That is microtubule. In that target, what is done? By inhibiting the microtubule synthesis, what will happen? Cell division will be inhibited. So growth of the bacteria, growth of the fungus will be inhibited. And the drugs which are used are griseofulvin. Drug that is used is called griseofulvin. Next, target number 5, and that is the last target. In that, what is happening? DNA, that is anti-metabolite drug. In DNA, what is happening? It inhibits the synthesis of DNA, and by inhibiting the synthesis of DNA, the drugs which is used is called flucytosine. Flucytosine is also called nucleoside antifungal agent. So by using the nucleoside antifungal agent, what is done? The growth of the fungal cell is inhibited. Now I will show you in diagram again, then it will be very much easier for you to remember. Okay? Just you need to remember in diagrammatic view, that is most important. Once you will understand the diagrammatic view, very easily you can understand or remember the drugs. Okay. So for example, this is a fungal cell. Where? See, this one is the fungal cell. Draw a cell. Is it visible or not? It is not visible. So this is the cell membrane. Outside of that cell membrane, what will be there? Cell wall will be there. And you know, I have mentioned earlier that all the cell wall are made up of sterols. In the case of mammal, the sterol is, in the case of mammal, the sterol is cholesterol. Cholesterol. And in the case of fungus, the sterol is sterol. In the case of mammal, it is cholesterol, and in the case of fungus, the sterol is ergosterol. What is that? ergosterol, and these ergosterol are placed there as a cross link from like this. So these all are ergosterol. Okay. So this is the target one. You can write T1. T1. Okay. Next, what will happen? You will see T2. So here is the T2. T2 is formation of ergosterol. Ergosterol is synthesized from squalene. Squalene to squalene epoxide. And that squalene epoxide is again transformed into lanosterol, and then again from lanosterol ergosterol is synthesized. So what will happen? Target number 2 is there, squalene epoxidase enzyme inhibitor. So in the squalene epoxidase enzyme inhibitor, what are used? Allylamine, that is T2. And what are the drugs? Allylamine. And example of allylamine. What are the example of allylamine? They are terbinafine, tolnaftate and naftifine. Terbinafine, tolnaftate and naftifine. Right? Next you will see T3 here. T3. Target 3. Target number 3 here. So in that what will be there? Latanosterol 14A demethylase. And latanosterol 14A demethylase is the enzyme which is inhibited by azoles. And what are the example of azoles? Miconazole, fluconazole, voriconazole, farconazole, treconazole, all the nazoles. Okay. Next, target 3. Next, target 4 will be there. See, there will be the nucleus. In that DNA will be there, and microtubule will be there. Right. So these microtubules are the target 4. And what are the drugs used here? Griseofulvin. Griseofulvin. Right? What are the drug used? Griseofulvin. Where? On the microtubule. And the last target, that is target number 5, that is DNA. And in that, what are the drugs used? Anti-metabolite, or you can say nucleoside antifungal. Nucleoside antifungal agent, and the example of flucytosine. Flucytosine. Okay? Example of flucytosine. Okay? Thank you so much. So up to this antifungal drugs, mechanism of action of antifungal drugs are completed. Hey, Uf Gaming, ভালো আছি